All right, let's talk about blood safety. If you're stepping into healthcare administration, quality assurance, or logistics, getting a grip on biological supply chain rules is an absolute must. It's not just checking boxes, it's literally the bedrock of patient safety. Today, we're taking those incredibly dense, highly technical federal requirements for human blood products and breaking them down into a practical, easy-to-follow roadmap. Let's get into it.
Here's the game plan. We're going to hit the blood safety rulebook, how we protect the human source, safe collection and processing, the crucial cold chain, quality control and testing, and finally, the real cost of noncompliance.
So think about this for a second. Every 2 seconds, someone in the US needs blood. That's a massive logistical hurdle. How exactly do we build a totally fail-proof supply chain when our raw material is living, degradable human tissue? It takes an airtight regulatory setup to make sure these life-saving therapies never turn into life-threatening hazards. Unlike building electronics or stamping out car parts, every single second matters here, and the environment has to be absolutely perfect.
Starting us off at number 1, the Blood Safety Rulebook.
Just look at this list. We're tracking a huge variety of materials, and each one comes with its own biological ticking clock and strict processing rules. See, the regulations treat a bag of whole blood completely differently than, say, separated red cells or purified proteins like albumin. As a compliance pro, you've got to realize that a one-size-fits-all approach just doesn't work. Every specific component has a custom-tailored regulatory pathway to guarantee it stays safe and effective for the patient.
Moving on to Section 2, protecting the human source, because it all actually starts with donor eligibility.
You know, the regulatory journey kicks off way before a needle even touches an arm. We simply can't guarantee a safe final product unless the person giving the raw material passes a super rigorous, highly documented health screen. Facilities are legally required to dig into a donor's medical history, physical health, and any potential exposure to infectious diseases. If someone doesn't meet those rigid criteria, they have to be turned away. There is absolutely zero wiggle room here, because letting a compromised donation slip through puts vulnerable patients right in harm's way.
To give you an idea, regulations strictly limit automated platelet donations to just 24 times a year. Why? To prevent donor depletion and give their bodies plenty of time to safely recover. There are also really specific timelines for how many days you have to wait between sessions, depending on exactly what and how much you gave. It's a great reminder that these rules are actively protecting the donor's health, not just the person receiving the blood down the line.
That brings us to part 3, safe collection and processing.
By mandating a totally closed, sterile loop, meaning medical-grade sterilization of the site, extraction through a completely sealed system, and immediate temperature isolation, we basically eliminate the chance of random outside bacteria ruining the product before the processing even starts. Regulators don't mess around here. Containers have to be sterile, pyrogen-free, and hermetically sealed. Any exposure to the outside air? Huge red flag. It's treated as a major contamination risk.
And let's talk about absolute traceability. If a problem pops up weeks down the road, facilities have to be able to instantly link that exact unit right back to the specific donor's medical file. This kind of tracking is really the backbone of biological compliance. Every single tube, bag, and sample has to have an unmistakable ID. Because think about it, if an infectious disease shows up in a donor's future blood test, the facility needs to sprint back to the freezer, find any previous donations, and pull them from the system immediately.
All right, Section 4, the crucial cold chain and temperature control. Blood components are incredibly delicate, and different parts need wildly different environments just to survive. So using a standard one-size-fits-all refrigerator is a massive compliance violation. You literally cannot just toss these bags into a standard hospital cooler. The law dictates totally different thermal environments based on exactly which piece of the blood you're trying to preserve.
This is exactly why facility managers obsess over climate controls. Check this out. Red blood cells need to be kept just above freezing, but platelets, they need room temperature. Plus, they have to be constantly, gently rocked back and forth so they don't clump up. Keep platelets too cold, and you destroy them. Meanwhile, source plasma that's going to be used for manufacturing has to be locked away in a deep freeze at negative 20 degrees Celsius or even colder. If plasma warms up, its manufacturing potential is totally ruined.
And it's not just about hitting the right temperature. It's about how fast you get there. For certain plasma products, the clock is ticking aggressively. Miss a regulatory freezing deadline, and that material either gets downgraded or goes straight into the trash. Say you're making fresh frozen plasma. The rules usually say it needs to be separated and deep frozen within 8 hours of collection. Staying compliant here means you need a flawless, heavily choreographed handoff from the donor chair to the lab, right to the freezer.
Which leads us to Part 5, Quality Control and Testing.
Here's the thing. You can't just trust that the system worked. You have to prove it. And that means running samples from every single batch through a gauntlet of tests before it ever gets cleared for medical use. The rules actually require multiple redundant checks. For whole blood, you're verifying ABO blood types and RH factors using totally different methods just to make sure there is zero mix-ups. You've got intense screens for infectious diseases, and it's not just lab stuff. Staff literally have to visually inspect the bags for weird colors, cloudiness, or broken seals right before they hand them over.
When we get into manufacturing specific liquid therapies, regulators start demanding exact chemical ratios. For instance, requiring a minimum of 83% pure albumin just to guarantee it actually works for the patient. If a facility is pulling specific proteins out of plasma, compliance gets incredibly scientific. The final solution has to hit strict pH levels, sodium limits, and precise protein makeups. It's all about making absolutely sure the patient gets exactly the right medicine with zero dangerous impurities.
And to knock out any invisible viruses in these protein therapies, the rules mandate a super precise baking process, exactly 60 degrees Celsius for 10 to 11 hours. Look at how incredibly tight that window is. If you heat the albumin for 9 hours, you're out of compliance and it's not considered safe. If you heat it for 12 hours, you might actually damage the proteins. The regulations demand meticulous, heavily documented precision at literally every step of the process.
Finally, Part 6, the cost of non-compliance.
So what happens when things go wrong? Well, regulators show absolute zero leniency for sloppy temperature control. An accidental thaw means immediate quarantine, and it could mean thousands of dollars of life-saving material just tossed in the garbage. If plasma accidentally warms up past the legal limit, you can't just stick it back in the freezer and pretend it's fine. It has to be extensively documented. Sometimes it might be heavily restricted and get slapped with a giant warning label. But honestly, most of the time, severe deviations mean total destruction of the batch. Documenting these kinds of failures is a massive part of what a compliance professional does.
At the end of the day, the whole point of every single regulation is to engineer a system that actively prevents contamination. These rules, they aren't just bureaucratic red tape designed to annoy you or dodge fines. They act as an invisible shield, protecting patients when they are at their absolute most vulnerable. Every single time you check a freezer log, verify a donor record, or audit a sterilization process, you are actively keeping patients safe. The protocols we've been talking about were literally written in the blood of past mistakes, and they exist to make sure those mistakes never happen again.
Because precision in the supply chain directly equals safety in the hospital bed. So as you look at your own facility's operations, and as you grow in your compliance career, I want you to ask yourself this. How will your dedication to these supply chain rules safeguard a patient's life tomorrow? Take what we've covered, apply it rigorously, and never forget the real human impact behind the rules. See you next time.
If you found this helpful or want to stay updated when this regulation changes, subscribe and support the channel. And if you need the full technical details on this CFR, click the link in the description below. See you next time.